Hi everyone, my name is Carrie Jansen. I'm an MD-PhD student at Emory University in Atlanta, Georgia. I'm going to talk to you today about our paper which detailed CD8 T cell infiltration into renal tumors. So as many of you know, in the literature, tumor-infiltrating T cells have been associated with a survival benefit in many cancer types. Tumor-infiltrating lymphocytes have been associated with improved overall survival, disease-specific survival, and progression-free survival in multiple tumor types. And you can see here in the excellent work done by the Galone group that the density of tumor-infiltrating lymphocytes in colorectal cancer specifically was shown to be a significant predictor of both disease-free and overall survival. And so based on this promising work and that seen in other cancer types, our lab was really interested in the question of what is the prognostic significance of tumor-infiltrating lymphocytes in renal cell carcinoma. So to do this, we obtained intraoperative samples, which we then processed and analyzed by flow cytometry and other laboratory methods. In the cohort of patients that we studied, these could be patients with any stage disease undergoing partial or radical nephrectomy before receiving any systemic therapy. And we received the tumor specimen at the time of surgery, which we then process and bank for biological analysis. So when we looked at the CD8 T cell infiltration in these surgically resected renal tumors, we observed a remarkably large variation in the CD8 T cell infiltration, where some patients have virtually 0 T cells in their tumors, while others have up to 25% T cells in their tumors. And interestingly, when we stratify patients by those with high or low CD8 T cell infiltration, we find that patients with poor CD8 infiltration progress much more quickly than those that have a strong T cell response. So the patients that have very many T cells in their tumor are surviving much longer without their disease recurring or getting worse, whereas the patients that lack this kind of immune response tend to experience worsening disease much more quickly. So an obvious next question was that we wanted to see if there were any other prognostic factors that might explain this difference in survival that we were seeing. And so I'm just showing a sample here, but when we examine a number of other characteristics of these patients and of their disease, we find that a patient's CD8 T cell response does not correlate with the tumor size, the disease stage, the disease histologic subtype, or with patient age, sex, or race. Another question we had was whether this response would correlate with PD-L1 positivity in the patient's tumors, and so we stained for PD-L1 and had clinical pathologists score the staining as positive high, positive low, or negative. And when we compare those tumors with high, low, or negative staining, there's no difference in the amount of CD8 T cell infiltration in any of these groups, and there's no significant difference in survival between patients with PD-L1 high, low, or negative tumors. And so given all these lack of correlations with these additional clinical factors that we saw, this really further prompted the question to us of what accounts for this variation that we see in the T cell response in these patients, and what mechanisms maintain this response. So to answer this question, we build upon the work of others in the field who have defined the stem-like model of the T cell response. In this model, there's a stem-like CD8 T cell that's defined by high expression of co-stimulatory molecule CD28 and the transcription factor TCF1, and low expression of checkpoint and effector molecules. This stem-like cell can both self-renew as well as differentiate into terminally differentiated cells, which lack expression of that transcription factor TCF1 and have high expression of checkpoint and effector molecules like granzymes and perforin. Importantly, many other groups have shown that these stem-like cells are also those that are critical for the response to anti-PD-1 type therapies. So when we looked in our human cancer patients, we can indeed find these stem-like and terminally differentiated CD8 T cells in the tumor. Here we see those PD-1 high, TIM3 positive, terminally differentiated cells in red, and then in green, the TIM3 negative, CD28 positive stem-like cells. Here again, we can see that the terminally differentiated cells have higher expression of those checkpoint molecules, whereas the stem-like cells have that characteristic expression of the transcription factor TCF1. Here we see by RNA-seq a similar result. The stem-like cells characteristically express TCF7, but the terminally differentiated cells lack this expression have much higher checkpoint molecule expression, higher expression of those effector molecules. Importantly, we can also sort these two cell populations out and stimulate them in vitro. And what we see is that while the terminally differentiated cells are unable to proliferate, the stem-like CD8 T cells undergo many rapid rounds of division, suggesting that these stem-like cells are those that with the proliferative potential to sustain the anti-tumor T cell response. Importantly, as these stem-like T cells divide, there's also an increase in expression of checkpoint molecules such as TIM3 and CD244 shown here. And this suggests that the stem-like CD8 T cells are truly giving rise to those terminally differentiated cells. And so here we're able to appreciate that differentiation both by examining the increase in these checkpoint molecules expression in vitro, as well as comparing checkpoint molecule expression in vivo. And so seeing this in vitro differentiation, this suggested to us that there is indeed a lineage relationship between these two cell types as had been explored in various mouse model systems, where this stem-like CD8 T cell is giving rise to those terminally differentiated cells. So to further explore this lineage relationship, we wanted to really try to confirm that in a different way by using T cell receptor sequencing of these two populations. So here we have T cell receptor sequencing results from a number of renal cell patients. On the left, each of these dots represents a unique TCR clone, where the green dots are those only found in stem-like cell populations. The red dots are those found only in terminally differentiated cell populations, but the dots in purple are those shared by both stem-like and terminally differentiated CD8 T cells. And this shows a very strong overlap between those terminally differentiated and stem-like CD8 T cell repertoires in these human renal cell carcinoma patients. On the right, you see this relationship at an individual patient level, where most patients indeed have that very strong overlap in TCR sequences between the two populations. Again, confirming this important relationship between the stem-like and terminally differentiated cells. And so since we've observed this stem-like CD8 T cell that gives rise to the terminally differentiated cells in the tumor, we can see that the amount of total T cell infiltration in the tumor also correlates with the amount of stem-like CD8 T cells in the tumor. Put differently, this means that losing the stem-like T cell in the tumor also correlates with the loss of the T cell response. And so then the question really becomes, so again, losing that stem cell means we lose those terminally differentiated cells and we lose the T cell response. So then our question really becomes, what maintains the stem-like CD8 T cell population? And so at the same time we were looking at these cells, we were also studying other immune cell types in these tumors. And we found that when we looked at patients with high, intermediate, or low T cell infiltration, the dendritic cell infiltration paralleled the variation that we saw in the T cells. So when a patient has very many T cells, they tend to also have very many dendritic cells. Conversely, when a patient has very few CD8 T cells, they tend to have very few dendritic cells as well. And so here you see summary plot of a number of patients. And interestingly, the same relationship did not exist when we looked at macrophages shown here or a few other immune cell types. And to us, this suggested that this relationship between the CD8 T cells and the dendritic cells was reflecting some sort of specific biological interaction between these cell types. So then the next thing we wanted to do was look at how these cell types, dendritic cells, stem-like CD8 T cells, and terminally differentiated T cells, were interacting in the tumors. And so to do that, we started doing some immunofluorescence imaging, but we also wanted to find a way to quantify that imaging data that we collected. So with the methods we used, we are able to take immunofluorescence data and extract a variety of quantitative measures. We can measure the XY location of each cell, as well as the fluorescence intensity of each of the markers stained for. So that allows us to plot the XY locations of various cell types. So shown here, just an example, in human tonsils, CD4 and CD8 T cells, MHC class II expressing cells. And we can also look and plot the co-expression of various molecules as well. So in this plot, we show those CD4 and CD8 T cells that are also expressing PD-1. And so then we can put all this back together and create a digitized version of the image that we collected. This digitized image, as well as the data that supports it, allows us to take many more measurements and quantitatively assess the biology that is reflected in this immunofluorescence data. So then we apply these quantitative methods to imaging renal tumors. Here we see CD8 T cells in red, where the stem-like CD8 T cells are those with the yellow centers, representing TCF1 expression. We use MHC class 2, shown in green, to mark antigen-presenting cells. If we zoom in here, we see dense aggregations of immune cells within the tumor. And we notice that the stem-like TCF1 positive stem-like cells really tend to reside in these dense aggregations nearby to densely clustered antigen-presenting cells, whereas we see the terminally differentiated cells more dispersed throughout the tumor. So if we use our quantitative methods to more concretely describe what we're seeing in this image, we can create a sort of immunomap of the tumor, where we have a contour here in gray of the density of the MHC class 2 positive antigen presenting cells in the tumor. And then we can overlay that with the XY locations of the stem-like CD8 T cells in green and the terminally differentiated T cells in red. Again, noticing how these stem-like CD8 T cells really seem to reside more preferentially in that antigen-presenting niche in the areas of high antigen-presenting cell density. On the right, I'll show you some additional measures to demonstrate this sort of immunologic niche that we're describing. Here we show that the TCF1 positive stem cells preferentially reside in areas of higher antigen-presenting cell density. And then we also show that the TCF1 positive cells, again, here correlate with the higher MHC class 2 density, and that TCF1 positive cells are on average much closer to their nearest antigen-presenting cell neighbor than the TCF1 negative cells are. So then if we take these quantitative methods of describing this stem-like cell residing within antigen-presenting niche up to look at large pieces of tumor, this will allow us to get an idea of what the global immune infiltration looks like within a patient's tumor, as well as to compare what the immune infiltration looks like in patients with and without progressive disease. So on the left here is an example of a patient with stable disease for greater than 2 years, while on the right we have an example of a patient that progressed rapidly after surgery. In the patient with stable disease, we can zoom in and appreciate these same antigen-presenting niches in the tumor. There are many regions of antigen-presenting cells, CD8 T cells, and importantly, many regions where both antigen-presenting cells and CD8 T cells co-localize, shown here in green. Conversely, the rapidly progressing patient, while there are some infiltrating antigen-presenting cells and T cells, this patient clearly lacks the same level of immune infiltration and critically lacks those regions harboring both antigen-presenting cells and T cells that are critical for maintaining that stem-like cell population. And when we measure the antigen-presenting cell density in a number of patients' tumors, we can see that patients without progressive disease have a significantly higher proportion of their tumor tissue with high antigen-presenting cell density, whereas patients with progressive disease lack this same level of antigen-presenting cell density. Similarly, if we stratify patients with antigen-presenting cell dense or sparse tumors, we find that patients with antigen-presenting cell dense tumors have significantly improved progression-free survival. Put simply, this means that the loss of the antigen-presenting cell zones or the absence of them could be occurring during tumor escape, and that these antigen-presenting niches that we're describing are really critical for maintaining the anti-tumor T cell response that we've shown to helps control disease and promote patient survival. So as we begin to appreciate the importance of this immune organization in tumors, a natural next question was to explore how this might relate to other phenomena of immune organization, such as tertiary lymphoid structures that have been described by others in the setting of both cancer and infection. So we took slides from our patients in our cohort and looked to see if we could find those histologic structures known as tertiary lymphoid structures. These structures are defined as areas of immune organization that are found in peripheral tissues and possess important characteristics such as distinct T and B cell compartments, reactive germinal centers, and recognizable high endothelial venules, just to name a few. These are very large structures identifiable here, even at our lowest magnification. And so while only a minority of our tumors had a significant presence of tertiary lymphoid structures, interestingly, we are able to identify these tertiary lymphoid structures in patients with either very high or very low CD8 T cell infiltration, as well as to appreciate that there are patients that do have very high CD8 T cell infiltration that also lack appreciable presence of tertiary lymphoid structures in the tumor. So in the top here, you see an example of a patient with high CD8 T cell infiltration and the presence of tertiary lymphoid structures. In the middle, a patient with low CD8 T cell infiltration and the presence of tertiary lymphoid structures. And on the bottom, a patient without tertiary lymphoid structures, but with a very high proportion of CD8 T cells in the tumors. And here you can see that when we quantify this with flow cytometry, we found no difference in the CD8 T cell infiltration between patients with and without tertiary lymphoid structures. We can also look at these tertiary lymphoid structures by immunofluorescence. So here we see a tumor on the top with tertiary lymphoid structures and on the bottom without. And we can compare the features of these tertiary lymphoid structures to those antigen-presenting niches that we've described. And so here we notice that the antigen-presenting niche seems to be quite CD8 T cell heavy when compared to the tertiary lymphoid structures that we found in these tumors. And so while it seems that these niches overlap in some capacities with the structure and function of the tertiary lymphoid structures, they don't seem to quite fit the definition that's been accepted of these tertiary lymphoid structures. They're much smaller and they have a seemingly different shift in their cellular composition. But interestingly, if we compare these antigen-presenting niches shown here on the top to functional areas within secondary lymphoid tissue, we find that they much more closely resemble the T cell zone shown here as an example from human tonsil. And so what we're starting to see here is that this coordinated organization of immune cells seems to be a really critical component of a productive T cell response. Whether in lymphoid tissue or in the periphery, there has to be a place for these stem-like CD8 T cells and the antigen-presenting cells to come together and organize to maintain a productive immune response. In other words, this deliberate organization of immune cells is required for a productive immune response. When an immune response is carefully organized, the stem-like CD8 T cells thrive and survive, a strong anti-tumor T cell response results, and the patient does well. But when the immune response is disorganized, there's no place for these stem-like CD8 T cells to live, and the anti-tumor T cell response fails. So the model then here that we're proposing is that in tumors with a productive immune response, there are areas of high antigen-presenting cell density that serve as homes for these stem-like CD8 T cells that sustain the anti-tumor T cell response by giving rise to new terminally differentiated cells. Conversely, when this antigen-presenting cell dense region is lost or absent, the stem-like CD8 T cell population in the tumor is lost and the anti-tumor T cell response fails. So in conclusion, we've shown that CD8 T cell infiltration predicts progression in human renal tumors. The CD8 T cell response is maintained by a stem-like CD8 T cell population that resides in these dense antigen-presenting niches in the tumor. And finally, the absence or breakdown of this deliberate immune organization is associated with the failure of the T cell response in the tumor. So, of course, I'd like to end by thanking my mentor, Dr. Hayden Kissick, as well as all the members of our lab, our collaborators in the Rafi Ahmed lab, all of the wonderful physicians that we work with at Emory's Winship Cancer Institute, our collaborators in the Sawalski lab, as well as the core resources that support our work, and of course all of the patients and their families who have donated their samples to make this work possible. Thanks so much for listening. Feel free to reach out with any questions.